Hi everyone, my name is Marco Zalatoye. I am a postdoc in Dr. Alex's lab here at the University of Illinois, Urbana-Champaign. And I have the privilege of introducing Dr. Alex this afternoon. So Dr. Alex received his PhD in statistics at Purdue University in 2009, and afterwards, he went to Professor Ed Buckler's lab to work as a postdoc for a couple of years. And during this period, he co-developed the GAPIT software, which most of us have widely used before. And recently, he got a job here at the University of Illinois Urbana-Champaign as a professor of biometry. And last year, he also co-developed a new statistical software for mapping epistasis loci in traits. And this software is also very, very efficient. I have used it myself, and I can recommend it to anyone to also use it for mapping. And this afternoon, we'll be talking on the exploration of the impact of genetic architecture on the performance of GWAS and GS models, that is genomic association studies and genomic selection models. And for some of these traits, he used simulation pipelines to develop, to simulate traits with different genetic architectures with different underlying loci from qualitative to oligogenic and polygenic traits. And also, he compared different GS models that can be used to effectively predict these traits. So over to you, Dr. Alex.
Okay. Well, Marcus, thank you so much for that awesome introduction. And I'd like to thank all of you for attending this and being willing to listen to me give my talk. So I guess without further ado, I will begin. Okay, so basically I'm going to talk about two different projects that my lab has done in the recent past. One has to do with advanced genome-wide association study models, and another one has to do with exploring the performance of a genomic selection model. So really briefly, what my lab does at the University of Illinois is basically we try to develop statistical approaches that associate the genotype to the phenotype. So the basic statistical model that is used is you can imagine your Y variable is a trait of interest, and then your X variables are one or more genomic markers. And so what we try to do is we try to develop and apply statistical models that can model the G2P, the genotype to phenotype relationship, as accurately as possible, and come up with genomic selection models that yield optimal, accurate genomic estimated breeding values. So the ramifications of the work that we do is that by developing more accurate statistical models that can encapsulate biological reality better, we'll be able to dissect the genetic sources of agronomically important traits. We can develop more flexible statistical models that can analyze a wider range of traits, like, for example, maybe discrete traits. And also we can, perhaps if we develop a really accurate genomic selection model, this will result in a reduction in the length of breeding cycles. So the first project I'm going to talk about, again, which is a GWAS approach, has to do with epistasis. And since I'm at the National Association of Plant Breeders webinar, I'm going to talk about dog color. So let's say that our phenotype of interest is color of Labrador dogs. We got black, brown, or yellow. And let's oversimplify a little bit. And let's say that we got a B locus that controls black versus brown. And we got an E locus that controls yellow versus not yellow. So in this picture up here, we see all possible 16 genotypes of the E and B loci. And then we have superimposed the observed fur color of a Labrador dog. So what we notice is that regardless of the genotype at the B locus, if you have a little e genotype at the E locus, your dogs are going to have yellow fur color. So this is a biological example of an epistatic interaction between the E and B loci. So, recent advances in computing power I argue, is now making it possible to go beyond the basic GWAS model where you test the association of one marker at a time and actually have models that can accommodate multiple markers and their interaction into a model, and thus be able to simultaneously quantify additive and non-additive effects underlying a trait in one statistical analysis. And so I believe that this approach, which was, for example, described here, developed for QTL analyses, can now be applied to big data, and it's a step in the right direction. So here's this model. Your Y variable is your phenotype of interest. Then you can do a stepwise model selection procedure to identify a set of markers that have significant additive associations and markers that have a significant two-way epistatic interaction with your trait. And then, of course, your model is going to have a grand mean and your error term. And so various model selection criterion can be used to help you determine the entry and exit criteria. So you can do an information-based criterion and a permutation procedure. And I'm just listing these here for FYI purposes. There's no pop quiz at the end of this slide. So basically, my research that I've done in this work has investigated the implementation of this procedure, and my colleagues who are computer scientists insisted that I come up with an acronym to describe this model. So what I came up with is SPAMEL. So if you hear the word SPAMEL in the next couple slides, I'm talking about this model. So what I worked on over the past 4 years or so was implementing SPAMEL into software that agronomical researchers can readily use and test it out in various data sets. So what I had to start off with is the Tassel suite of Java programs from the Buckler Lab, USDA ARS Cornell, where I did my postdoc. And basically, while I was a postdoc, I helped implement a stepwise model selection, SPAMEL, just for additive terms only, into the Tassel GUI. So I leveraged this, and I added some bells and whistles to the code so we could also do stepwise model selection for pairs of epistatically interacting loci. So this was a starting point for me to assess multiple testing correction issues. As the number of markers increase, the more difficult it is to identify the correct QTN. And also computational issues. If you're doing SPAMEL on 1 million genome-wide markers, it's going to take forever to run. And when I say forever, perhaps hundreds of years. So this preliminary analysis I've been doing was a confirmation of what has been shown in the literature. Models like SPAMEL are useful, but not on a genome-wide scale. So my future direction is to come up with biological approaches to help you hone in on specific loci in which to try SPAMEL, but more on that later. So this current study I'm going to talk about, which was published in Heredity, was basically to find out how good of a job does SPAMEL do with identifying quantitative trait nucleotides. And as Dr. Olatoye, my postdoc, was alluding to, we did a simulation study. So basically, we just took genome-wide markers, we randomly selected one of them to be QTN, and then that formed the genetic basis of our simulated traits. So what we did was this. We simulated phenotypes in 2 data sets. One was a maze diversity panel. For those of you familiar with maze diversity panels, this was the Ames panel. And also we looked at a human case control study. Both had between 2,000 and 2,700 individuals. And then we varied the number of markers in which to test SPAMEL on. So 5,000 versus 15,000. And again, we would randomly select these markers, a subset of these markers, to be QTN with either additive or epistatic effects underlying our simulated trait. We varied the number of individuals, sample size of 300 small, but we wanted to see how SPAMEL performed with small sample sizes. And then we did 5 different configurations of number of QTNs and their effect sizes, 2 of which I'll highlight in this presentation. One is based upon what is known in the literature about the genes underlying maize inflorescence. Here's the corresponding broad sense heritability we simulated. Then another one was based off of what is known in the literature about the genes underlying Alzheimer's disease in humans. So both of these have approximately 24 additive effect loci of subsequently smaller effect, and then 1 major large effect epistatic QTN. And then we compared the performance of spamel to 2 other approaches, one being joint linkage analysis, which is just basically stepwise model selection with no epistasis, and then also a canned package that exhaustively tests for every pair of available markers. So this is a massive study. If you want to see everything we did, check out the heredity paper. For the sake of this presentation, I'm going to focus on maximum number of markers, maximum sample size, these 2 genetic architectures. So the first question we wanted to ask was, well, hey, how good of a job does spamel do with identifying the QTN that we simulated relative to the 2 other approaches that we tested? So here's a really busy graph, a really busy picture showing these results. So our first column is the trait we simulated to mimic inflorescence in maize. And then this column shows results for the trait that we simulated that mimics what is known about Alzheimer's disease in humans. We simulated these data in both the human and maize marker data set. The first and third row show the results for the human data set. The second and fourth row show the results for the maize data set. The first 2 rows show the results for the additive QTN. The bottom 2 rows show the results for the epistatic QTN. For each graph, your x-axis is the effect size of your QTN, and then your y-axis is the proportion of times out of 100 simulated QTN that a given model identifies are QTN. So what we're seeing for the additive QTN is that as your x-axis increases, that is, as your effect size increases, the proportion of times that joint linkage analysis and spamel, which is testing for epistasis, increases at about the same rate. In a similar manner, for the epistatic QTN, spamel and joint linkage analysis have very similar positive detection rates, while fast epistasis never detects any additive QTN, and it has variable performance on the detection of epistatic QTN. So take-home message of this graph is that we are observing similar QTN detection rates between spamel and joint linkage analysis. That's great. Our next question we wanted to ask was, how good of a job does spamel do with distinguishing between additive and epistatic QTN? That is, if the truth is that our QTN has an additive association, how often does spamel consider it additive? If we have a true epistatic QTN, how often does it identify both loci as having an additive 2-way interaction effect? So the layout of these results is similar to the previous page. So if you look at the results for the additive traits, we see that for the human, the additive QTN, we see for the human data that as your additive effect size increases, this blue line is getting taller. So this is telling us that for the human data, as our additive effect size increases, we become more and more able to correctly identify an additive QTN as additive via spamel. Not so much for maize. We see that in the maize data, it's definitively misspecifying an additive QTN as epistatic. For the human data set, when we simulate the epistatic QTN, we see that spamel is capable of correctly identifying both loci contributing to the epistatic QTN and correctly calling it epistatic, not so much from the maze data. And then another thing to point out for this Alzheimer's disease-like trait, this locus over here, we simulated an additive and an epistatic effect there. So basically what this result is telling us is that spamel for the human data set is capable of distinguishing between an additive and a epistatic signal at the same locus. So to conclude, in the human data set, spamel is able to correctly classify an additive QTN as additive and identify both loci contributing to a two-way epistatic QTN. So why is spamel performing so much better in the human data set? Here's a result that potentially could lead to a hypothesis, namely that it has to do with minor allele frequencies. So here on this x-axis, we're looking at the distribution of minor allele frequencies for the markers we considered in the human data set, and here's the MAF, so the markers we considered in the maze data set. So here, we're seeing that we have a much more prominent distribution of common alleles, non-rare alleles in the human data than we do in the maze data. Incidentally, these purple dots are the minor allele frequencies of the loci we randomly selected to be QTN. So some conclusions. How good does spamel perform? Well, spamel is as good as joint linkage analysis for detecting QTN. However, correct specification of additive versus epistatic QTN are associated with minor allele frequency. So here are thoughts about moving forward on this project. We are currently collaborating with folks to develop a priori methodologies based on either a priori biological knowledge or maybe machine learning or other approaches like that to prioritize specific genomic regions to apply spamel, okay? So this will help facilitate, this will help reduce multiple testing correction and computational issues because you're considering subsets of the genomes in which to apply spamel instead of the entire genome. Another thing we're going to try to do is migrate spamel to a lower level programming language. My unqualified opinion, Java is awesome. However, it's not as fast as other packages. So, programming languages. So I'm collaborating with the National Center for Supercomputing Applications here at University of Illinois with some experts who have the capabilities to move this to a lower level programming language like Spark or Apache, parallelize the code, and make it really quick and computationally efficient. My ultimate goal in this research is to have a bunch of statistical tools that researchers do not need to have a master's degree in computer science to be able to run them and be able to test for epistasis in computationally efficient software. So that's my first project I'm going to talk about. My second project has to do with genomic selection, and I was debating whether or not to give some introductory slides to genomic selection, and so I kind of took the middle road and I gave not my full introduction to genomic selection, but just a couple of slides. So basically, the purpose of genomic selection is we want to predict genomic estimated breeding values, or to oversimplify, phenotypic values using a genome-wide set of markers. So basically what you're going to do is you're going to take a training population that you genotype and phenotype for your traits of interest, and you train a genomic selection model. Your Y variable is your phenotype of interest, and your X variables are your genome-wide set of markers. So you fit that model to these data, and then you go into breeding material, which are genotyped with the same markers. And basically, you plug in the marker genotypes into the fitted genomic selection model, and you calculate these predictive phenotypic values, more technically speaking, GEBVs, genomic estimated breeding values. And then you make selections based off of these GEBVs. So these enable crop breeders, to select without having to phenotype individuals. So if you imagine that, you don't have to go through an entire field season to wait for your plants to phenotype, or if you're planting perennial species, you don't have to go through a couple of, burning years or whatever to obtain your phenotypes. You can make selections on the spot as soon as you sequence your breeding material and calculate these GBVs. So to oversimplify even more, various frequentist, i.e. non-Bayesian and Bayesian models are commonly used for genomic selection. In my experience, and the literature seems to agree with me on this, that most produce approximately the same prediction accuracies. My second introductory slide I'm going to show is, what happens if you have only one data set and you want to see how good a particular genomic selection model performs. Well, here's one commonly used approach to assess how good of a job a genomic selection model is performing. And this is cross-validation. So what we have over here is 5 different squares. So what this represents is a data set, a diversity panel, or a breeding population, if you will. And it's subdivided into 5 approximately equally sized subsets of individuals, okay? So let's say we partition the data into k-folds. Well, here is k-folds where k is equal to 5. So, so 4 out of the 5 of the folds are going to be your training set. So you fit your genomic selection model into your k-1 folds. And then in your test set or your validation set, you plug in the marker genotypes into this fitted genomic selection model, obtain your genomic estimated breeding values, and then what you're going to do is you're going to calculate the Pearson correlation between your observed and predicted trait values. And then you repeat this procedure so that each of these squares gets to be red. That is, each square gets a chance to be the validation set, the test set. And then your prediction accuracy, one way of measuring this is your average correlation between your observed and predicted trait values across folds. And then the closer your correlation is to 1, to positive 1, the higher your prediction accuracy is. Okay, so the particular statistical model that I'm going to talk about here happens to be one of the most widely used, arguably the most widely used statistical model for genomic selection. So your Y variable is your phenotype of interest. And then for each of your markers, let's say we have P markers distributed throughout the genome. Each marker is going to be fitted in your model as a random effect. And then you have your error term and you have your intercept. The predictions, the blocks of this random marker effects are subjected to this ridge regression penalty over here. And so an important thing to point out here is that all predicted marker values from this models are equally penalized. That is, they are equally shrunk to 0. So no matter the size of the genes, no matter the effect size of the genes they are tagging, the predicted mark values are going to be equally shrunk. So here's a picture of my PhD student, Brian Rice. And this work is what he did for his master's degree. So he looked at the literature, for example, Bernardo's paper from 2014. Let's suppose we have a complex trait, hundreds or thousands, hundreds or thousands of small effect loci combined with several large effect genes. Is this penalty of the R or BLUT model, is it unfairly shrinking the prediction, i.e. the effect size of the markers tagging these large effect genes? And are we getting an underestimation of our prediction accuracy accordingly? So random effect markers have this ridge regression penalty applied to their predictions. But if you have a subset of markers that are also included as fixed effect covariates, they don't have this penalty. So Brian's research question for his master's degree was, if we have a certain subset of markers that we include as fixed effect covariates, can we increase the predictive ability of over a traditional R BLUT model? Can we get higher correlations between observed phenotypic values and genomic estimated values? So we compared the predictive ability of a traditional RR blood model that I showed a couple slides ago versus an RR blood model that has a fixed effect marker covariates. And we selected these fixed effect marker covariates in an elaborate manner that I'll explain in a second. And we did this across a wide range of genetic architectures for simulated traits. So what we did was this. We simulated data in a maize diversity panel. This is the Goodman-Buckler diversity panel, and in a sorghum diversity panel. Both have approximately 300 individuals, and both have in the ballpark of 50K to 90K markers. And so similar to the spamela simulation study, we randomly selected a subset of these markers to be quantitative trait nucleotides. And then we simulated our traits in two different configurations. One is a classic complex-ish kind of trait where you had QTN effect sizes that got progressively smaller and smaller and smaller. And then our second type was the genetic architecture that we hypothesized that including these fixed effect covariates could boost prediction accuracy, namely where we had one large effect QTN and then a whole bunch of really small effect QTNs. And then a user inputted their selected narrow sense heritability, and we obtained our simulated trait values accordingly. So here's the elaborate pattern that we did to evaluate prediction accuracy. For each simulated trait in each species, we would run GWAS in the training set, and then we would assess the predictive ability in the validation set of an R blood model plus a predetermined number of peak-associated GWAS markers from this GWAS. And for each simulation setting in each trait, we will repeat this procedure 50 times. Now, our expectations going into this study was to see, maybe half the time, maybe most of the time, this model over here, where we include these fixed effect covariates, would boost your prediction accuracy over a standard RR blood model. And boy, were we wrong. Incidentally, if you want to read this entire study, check out our plant genome paper that was published in March. So here's a graph of results. The take-home message is that more often than not, we did not see an increase in prediction accuracy. So here are the results of our simulation studies, subdivided into whether we were simulating our data in maize, and whether we were simulating our data in sorghum, whether we simulated a standard complex trait versus simulating a complex trait with a large effect QTN thrown in. So the x-axis on each of these graphs are the number of markers that we included as fixed effect covariates in our RR blood model. And then the y-axis is the increase in prediction accuracy over an RR blood model with no fixed effect covariate from an a priori GWAS. And just to emphasize that we mostly saw a decrease in prediction accuracy after including these fixed effect markers, here is a line at a y-coordinate of 0. So seeing the bulk of the times that we simulated traits, we see a decrease in prediction accuracy. So including a fixed effect covariate more often than not decreased prediction accuracy over a standard RR blood model. But it gets even better. So we also found out that including a fixed effect covariate not only does it more often than not decrease and increase prediction accuracy, it also increases the variability in prediction accuracy compared to a standard RR blood model. So here's a prediction illustrating one instance where we saw such a result. So here's a trait simulated in maize, a type 2 trait, I believe, with 10 QTN, an effect size of the large effect QTN of 0.5, narrow sensitivity 0.5. X-axis is number of markers included as a fixed effect covariate, and then the y-axis is our prediction accuracy across our 50 replicate traits. And indeed, we see relative to a standard RR blood model, which is depicted here at x-coordinate of 0, we are seeing the bulk of the prediction accuracies are increased relative to standard RR blood model, but check out the IQRs, the whiskers, and the range of possible values of these bug flies. we see a much wider range of values of prediction accuracy for a given number of fixed effect covariates relative to an R blood model. So another potential danger of including these fixed effect covariates for these given sample sizes is we can see an increase in variability in our prediction accuracy. The third thing that we noticed was this. In an ideal situation, we would like to see no bias in our genomic estimated prediction values. That is, let's say we are looking at plant height in maize, okay? Let's say that you fit a genomic selection model and you yield really good prediction accuracies, maybe of like 0.8, and I'm just making up that number. However, let's say that the range of values for your trait, say plant height, is between 30 meters and 100 meters. That's not very realistic. So, that's an example of a biased, predictions from your GEBVs. So what we did here is we illustrated an instance where we see an increase in the bias of our predictions after including a fixed effect covariate. So this is just from one trait. So this row over here, we see 0 fixed effect covariates, and I'm going to focus on the slope. I forgot to mention. So what we're doing here is we're performing a simple linear regression where your y variable is observed trait value and your x value is your GEBV. If you have no bias in your predictions, you would expect to see your intercept estimate to be around 0 and your slope estimate to be around 1. So here, we have no fixed effect covariates, a standard RR blood model. Sure, our intercept is a little different from 0, but our slope is pretty darn close to 1. When we have one fixed effect covariate, our slope estimate jumps up, suggesting an increase in bias. And then when we keep on adding these fixed effect markers, the bias generally goes down. Anyway, this is showing that including fixed effect covariates have a tendency to increase the bias of our predictions. Okay, so a conclusion from this study is that if you want to use genomic selection for your breeding program, the risks of including these fixed effect covariates may outweigh the benefits. So what we suggest doing is conducting a cross-validation study with data you have in hand, comparing a standard RR blood model to a model that has, GWAS results from a GWAS conducted in a training set, including fixed effect covariates, and look at how the prediction accuracies differ. Worst case scenario, no harm, no foul. This cross-validation study will tell you that having no fixed effect covariates is the best way to go, and you should proceed with the standard RR blood genomic selection model. Okay, so that concludes my talk. I'd like to acknowledge the current members in my lab, my collaborators at UIUC, and my collaborators throughout the United States and the world. And thank you all for your attention and for listening to this talk.
Alex, thank you very much and great job. We appreciate the information that you provided. And we would like to open this talk up to questions. So if any of you have a question for Alex about this topic or any related information, please enter it into the Q&A portal and we will get to it. So yeah, feel free to ask questions. We've got several minutes, and so we'll stand by for a few more minutes and hang out for questions. So thank you again, Alex. We really appreciate that.
No problem, my pleasure. So should I stop sharing my screen? Actually, never mind.
You're fine. You can leave that up, Alex. That's
okay.
Fine.
Okay.
So we did have a question from Anna, and she was asking about, so Alex, I think maybe a couple of slides back. So the prediction accuracy was higher when fixed effects QTL were used.
Yeah.
And then she mentions that they have higher variance, but looked at the values were higher, which is different from the slide before, where fixed effects decrease prediction accuracy. So are you able to locate those slides in question?
Yes, so this is on question 19, and right. So let me make sure I get the question down correctly, and then once I'm on the same page, I'll answer.
Right.
So this is a result for one of the genetic architectures we simulated. And in this particular instance, we see an increase in prediction accuracy. So I believe Anna pointed out that if we go to the previous slide, if I can get this thing to work. I was overzealous and I overshot it.
No problem.
Most of the time, we're seeing a decrease in prediction accuracy. So is the question about what's the discrepancy, or?
I suppose that's the case. Anna, could you provide, would you let us know if that's accurate?
Okay, I see, yes. What is the discrepancy? Okay, fantastic question, and thank you for that question, Anna. So, indeed, most of the time we see a decrease in prediction accuracy in our results. However, there are certain instances where we do see an increase in prediction accuracy. So, for example, if we go to these sorghum type 2 traits, we see some dots that are above the y-coordinate of 0, and these are instances where we see an increase in prediction accuracy. So this result I'm showing over here happens to be, let's see, I did it again, happens to be one of the instances in the simulated genetic architectures where we see an increase. Does that answer your question?
Alex, thank you for clarifying that. That's good information. And again, we have a few more minutes. So if you have any questions for Alex, we'd love to get you in here and address any of those. So Anna, yes, the variance is in the example represent the variance in other instances too. So Alex, are you able to see that?
Yeah, so let me, so the variance in the example represent the variance of the other instances too. Okay. Yeah, so the increased variance that we see here when we add a marker as a fixed effect covariate, we have observed that in other genetic architectures as well that we simulated. All right?
Excellent. Thank you, Alex.
And Anna and everybody else, we have this paper published in the Plant Genome. It appears in the March issue. So if there are any other questions about this work, please feel free to email me and or my my PhD student Brian Rice, who's the lead author, and we'll be thrilled to answer any detailed questions about our findings.
Alex, do you have a link to that paper?
Yeah.
Any abstracts to it?
Yeah, let me see. Let's see. Yeah. If you bear with me 1 second, let me go into Google Scholar.
So Alex is going to dig up the abstract for the paper in question, which gives us a lot of information, which he went into detail in on this talk. Again, we thank you for joining us today. We are going to post this on the Plant Breeding and Genomics YouTube channel in the next couple of days. And we do have a few more minutes for questions, so please get those in, and Alex will address them.
Okay. And I just put a link to the abstract of this paper up here. Is it actually the abstract? I think it is. Yeah, it's the abstract.
Fantastic. So I reposted that, Alex. I think it was maybe just to the panelists. But so yeah, so the abstract to the paper, which Alex has talked through, is now posted in the chat. So please look there. So Alex, we have a question from Valerio. Mm-hmm. Some of the models are non-parametric or Bayesian approaches available allow you to include variable QTL effects or known QTL effects. Mm-hmm. What would you expect to happen to accuracy in that case?
Okay, so models that include QTL effects, yeah.
Right. So models with non-parametric or Bayesian approaches with known effects, what would you expect to happen to the accuracy in that case?
Yeah, so I would say that if those models were applied to these data and these traits that we simulated, I would expect to see similar results. That is, yeah, including QTL's fixed effects, I think is going to have a similar impact on prediction accuracy, you know, and like variability in results, and also bias. What I think is going to happen for both the approaches we have tried in this paper and these other non-parametric approaches that include, you know, a priori QTL, is that as your sample size increases, I think that the variability, the variability that we see is going to decrease, and I think that the accuracy is going to increase. So, sorry, and I'm just thinking out loud here a little bit. So if like the QTL and or the a priori GWAS result is tagging a true causal mutation or is the true causal mutation, I would expect that as your sample size increases, models that include that causal mutation or something tagging it is going to see optimal prediction accuracies.
Excellent, Alex. Thank you for addressing that. Again, we have a few more minutes for questions, so please get those in and Alex will address those. Alex, I do have a question briefly. So a lot of what you focused on was maize, which is a major crop and deserves lots of attention. Have you seen similar results or maybe contrasting results in any different crops? Or has any of this type of work been done in, say, soybean or cotton or other crops that you maybe have come across?
Yeah. So yeah, I'll say really quickly that yeah. So we also investigated, for example, this GS study. We also investigated in as sorghum as well, and we see a little similar. Yeah, we basically have the same take-home message in sorghum as we had with maize. And right now, my lab is collaborating with people working on like miscanthus, some species which are polyploid. And in an effort to answer this question as concisely as possible, I would expect similar results in other species. And then with that said, I think it's important to test out these approaches in as many, you know, genetically diverse, or sorry, as many diverse crops as possible, so you can see how these approaches perform across the entire spectrum of genetics of crops.
Excellent. Thank you for that, Alex. So we'll hang for just another moment and address any more questions. So please get those in to either the chat or the Q&A, and we'll get to those. Again, if you have not registered for the NAPB annual conference, please do so as soon as possible. So the early bird registration ends on June 1st, and the meeting itself will be held August 25th through 29th in Callaway Gardens in Pine Mountain, Georgia. We hope to see you there. It's a great meeting. Alex, as we stand by here and wind down, do you have any more comments or thoughts that you'd like to add to the group?
Well, I'll just say thank you again all for listening. I will be happy to answer any questions, any follow-up questions over email. And if you all want to talk to me in person about anything I presented or anything pertaining to quantitative genetics, feel free to catch me at the NAPB meeting.
Thanks, Alex, and thank you all for attending today. We really appreciate this. This recorded webinar will be posted on the Plant Breeding and Genomics YouTube channel in a few days. We're going to end for today and hope to see you at the Plant Breeders meeting in August. All right, Alex, thank you very much.